I want to welcome everyone back to MVZ Lunch. And I'm sorry that we're still doing it by Zoom. I promise you this won't go on forever, although none of us can really predict when we'll be back in person. But I look forward to that day. Part of what makes us so great as a community is our interactions in person. So I appreciate everybody who's been coming back to work in person and interacting. I think we have the rest of the semester filled with seminars, so check the MVZ website to see who's speaking each week. Are there any announcements before I introduce Chris? Okay, well, it's a real pleasure to introduce our very own Chris Martin. Chris has led a peripatetic lifestyle in the last 20 years, bouncing principally between the East and California. So he did his undergraduate work at Duke, and I should say bouncing between the East Coast and California with side trips to the Caribbean and Africa, because after Duke, he was a Fulbright scholar in Africa and Malawi, and then came to Davis for his PhD. After that, he stayed in California and did a Miller postdoc with Craig Miller here in Berkeley before moving back to North Carolina, where he took his first faculty position as an assistant professor at UNC. He was there for 4 years, and then we were lucky enough to get him back to Berkeley in California, and so he's joined us and been here since 2019. So many of us have heard Chris speak before, but his lab is so active that he's always full of new things, and I'm sure we're going to hear new things today. He's been here for 2 years. As I said, he was at UNC for 4 years, and so he's coming up for tenure this year, and it's an especially fun time for us to hear about all of his latest research. He has wonderful projects on Caribbean pupfish that are federally funded. He has funding from both NSF as a career award, which is a real accomplishment as a new investigator, and from NIH. There aren't many evolutionary genetics projects on fish that are funded by NIH, to my knowledge. So that's a real accomplishment. He has active programs, as I said, on pupfish, but also on African cichlids. As the new curator of fishes in the MVZ, he brings a new vertebrate group to the MVZ, which we're very excited about. And in a short time here, has added 17,000 specimens to our collections. So he's been active in his curatorial duties as well. So I don't want to take any more time from you, Chris, but with that, I'll hand it over to you, and thank you for agreeing to speak to us today.
Thank you so much, Michael. That was a truly kind introduction, and I'm just incredibly honored and privileged and just humbled to be sitting here and talking to you all today. I mean, it really is just surreal, and I still just haven't even gotten used to it, especially with everything going on. So thank you all for coming. And so this is a great time to reflect on my career, and I have been studying these wonderful quirky pupfish for about a decade. I'm rounding down. But exactly 10 years ago, I published my first paper, and my first chapter of my dissertation was on these Caribbean pupfishes and their trace diversification rates through time. But also reflecting on my entire scientific career, exactly 2 decades ago, I met a real life scientist for the first time. I learned that a PhD takes more than 2 years. And I also, this photo was taken, and then 3 weeks later, I'm a millennial, right? So we're used to these decadal catastrophes. And so 3 weeks later, 9/11 happened. And then 1 year into my graduate education, there was the near collapse of global financial markets. And then, of course, now we're in this 18 months of a pandemic. And of course, I'm speaking to you now to myself, which is also surreal. But I bring all this up because we're back in the MVZ. Kind of take a deep breath. And this is this amazing community, as Michael mentioned, which is hopefully just a wonderful community and space to reflect and do our science. But I want to emphasize that, for me and for all of us, right, that this is actually, as biologists, I think that this is the response to what's going on out there, right? As our mission as a natural history collection and our sort of global challenge facing all natural historians is to go out there and to document what's out there, catalog and voucher what's out there as we meddle through these catastrophes. And earlier in my career, somebody explained this to me and described this to me as photographing the Titanic. This is our mission, is as natural historians, is photographing the Titanic. And of course, this is so much more than just photographs, right? These voucher specimens record that species at a particular time in a particular place, but also provide hopefully a record of genomic information and even a potential for genetic restoration in the future. So for me personally, the best response to what's out there and what I've seen every decade of my career so far is what we are all doing together. And of course, this has already started. In my first MVC seminar, I talked about the extinction of this pupfish. This is Megupsilon aperis. It's the sister genus to all other cyprinodon species, a monotypic genus. The last two males went extinct in my fish room when I was a postdoc at Berkeley. So this is actually my photograph of an endling, the very last individual of a species on Earth. And I still kind of carry this with me. This is this kind of profound sadness and sort of personal loss here with this species, right? There's so many scientific questions we could have asked about why does this genus, it was stuck in a little desert oasis in northeastern Mexico, and it failed to diversify for millions of years, whereas cyprinodon diversified across the Caribbean. And now the answers to that questions we'll never know. And all of us have stories like this of populations and species that we lost. Indeed, all the species that I'll talk to you about today are critically endangered or officially recognized as such as the Brombian bow cichlids. And I think most of these might go extinct during my career. So perhaps that's why I'm in a hurry most of the time. And so my lab studies these simple, sort of fragile ecosystems where we see the recent origins of adaptive radiation and speciation. We see abundant ecological opportunity in these new environments with the rare origins of novelty and adaptive radiation. And for my talk today, this is my third MVZ talk in 6 years. I'm really just going to focus on the accomplishments of my lab members over this time period and essentially just the highlights of this with some new stories and some old stories. And essentially, for the past decade, a decade ago, we thought we knew all of these things about novelty. In terms of their ecology, their behavior, the genetic basis of novelty, and the fitness landscape surrounding novelty. And it turns out that every single one of these assumptions is wrong. And so I'll walk through these throughout my talk and just point out just how weird novelty is. So what is novelty? Some of my favorite examples, my favorite definition comes from Supreme Court justice. Essentially, you know it when you see it, right? Like this leaf-nosed Malagasy snake, or this amazing horn on a unicorn fish, unicorn tang, which we still don't know the function of. We can also think of novelty as taking an existing structure and using it for a new function. So like the trichomes, our modified trichomes are these new mechanical traps on the Venus flytrap. We can also think about ecological novelty, which is the focus of my lab's research program. So here's the vampire finch, which is actually on 2 Galapagos islands, has started to feed on the blood of blue-footed boobies and some gulls. Just a sort of fantastic example of novelty. Of course, my favorite example is the scale-eating pupfish. So this is Cyprinodon desquamator. I named this species desquamator, which means one who removes the scales from fish. And there are independent evolution about 20 times of scale-eating fishes across the globe. But it is one of the rarest trophic niches among all marine and freshwater fishes. Here are some examples here. It has evolved numerous times within East African Rift Lake cichlids, such as these examples here, but there's no other scale-eating specialist among all other cyprinodontiform fishes, which is over 2,000 species, So this scale-eating pupfish is also the youngest scale-eating species on Earth. So 50% of their diet is the scales of usually generalist pupfish. And they're not just eating the scales, but the sort of protein-rich mucus coat on the sides of these fish. Here that is in the lab. This is filmed at 2,000 frames a second. Look at the spray of scales off the side of that fish. So first of all, we think about novelty as this unique thing that only happens once. But novelty is actually common within adaptive radiations. And I've actually argued that there's a third axis of adaptive radiation, not just species richness and phenotypic disparity, but also ecological novelty and ecological divergence. So we can think about things like pigapodid geckos and Burkinia bromeliads, which have extensive ecological novelty and sort of moderate amounts of phenotypic and species richness. And one of my favorite examples, again, of this is Barambiambo, Cameroon. Here's the lake from one end to the other. This is a crater lake, about 100 meters in depth, but incredibly simple environment around this crater rim, where 9 out of the 11 species in this radiation coexist. It's essentially just leaf fall and rocks. And yet we have many examples of ecological novelty within this system. So this is Pungu. This is a sponge-eating specialist, but one of only 2 freshwater sponge-eating specialists in the world that we know of, both of which are in Cameroon crater lakes. And many of these rocks are encrusted in freshwater sponges. But in the same lakes, we have a deep water specialist, this is Daikume, which basically hunts in and out of the anoxic zone for essentially only the gnat larvae that live there. This species never comes to the surface, and it actually has 3 times the hemoglobin concentrations of any of the other species. And then the third amazing species here, we discovered that this species is nocturnal. People thought it was extinct or rare. It turns out we've only found these at night, and it eats more shrimp than any of the other species in the lake. And of course, we see novelty again and again on San Salvador Island as well. So within these hypersaline lakes on San Salvador, we don't just see one scale-eating specialist. There's a second specialist here, which is the molluscivore. So here I'll show you these habitats. They're just dominated, again, simple environments dominated by macroalgae. Almost all the pupfish you see here are generalists, feeding mostly on little invertebrates and little macroalgae. At the very end, there's a scale eater. Here they are raised in the lab. They maintain this sort of very distinctive breeding coloration in the lab. And here he is recently. This is filmed on the 5th floor of VLSB at about 1100 frames per second. So you can see they're removing that mucus coat as well as the scale from this euthanized zebrafish. But in these same lakes, in the very same habitats, we see another specialist. And so this is the molluscivore, Cyprinodon brontothoroides. And here you can see its upper jaw here is actually tucked up underneath this large nasal protrusion. And so this is actually a skeletal extension of the maxilla here, as well as the nasal bones on this cleared and stained specimen. And here on a CT scan, you can see that very large extension of the head of the maxilla here in blue. Whereas in comparison, the generalist has these normal oral jaws. The scale eater has these massive oral jaws, about twice as large as the other species. And so we might ask, what is this nasal protrusion for? What's novelty for? And in one of my favorite teaching moments, Christy Dixon was in my undergraduate course-based undergraduate research experience class, right? And she was also in the lab. And she comes up to me and she says, Chris, I've been watching these eat, and I think that they pull the snails out of their shells. And I basically said, no way, prove it, right, essentially. And so a week later, Christy comes back and she shows me this video that she took on her iPhone. And there, you see it? So we followed up with this. Here it is at high speed. There's a molluscivore feeding on a snail. And he pulls it right out of their shells. So these fish are so small, they can't really crush the large snails found in these lakes, but they're sneaking up on the snails and essentially shaking them out of their shells. And so my fantastic grad student, Michelle St. John, who's now a 5th year student in the lab, but for her 3rd chapter, I believe, she teamed up with Christy and they looked at a whole bunch of F2 feeding trials. So here on the x-axis, we're showing size-corrected nasal protrusion distance in these F2 hybrids. And then on the y-axis is the largest snail that these fish were able to remove from their shell over a whole week. So it takes them a while to get around to it. And here in red is that line. But there's at least a trend that larger nasal protrusion is helping them extract the snails from their shells. And again, there's lots of other species out there with nasal protrusions, but no other cyprinodontiforms have this. And the closest ecological analog that I can find so far is this species. It's endemic to the coast of South Africa. Maybe Rory can help me find one day. It's a black mussel cracker, and it actually lives in these rocky reefs and feeds on mussels with this amazing nasal protrusion, right? Now, novelty is so common within adaptive radiations that we actually missed one. So in some of these lakes on San Salvador Island, there is a fourth species of pupfish coexisting in this lake, so it's still undescribed. And so my fantastic grad student, Emily Richards, who's now in her 6th year, she actually discovered this species using machine learning. So for her first chapter, she was using Saguaro software to essentially segment pupfish genomes into different phylogenetic histories. So as we kind of scan along the genome, we can see these different topologies. And what Emily kept finding is that across the dominant topology, across 64% of the genome, would separate out these sort of light blue clade from this dark blue specialized desquamator clade. And that was across lakes. And so initially for her first chapter, we said, oh, this is potentially a new species. But now Emily has followed up with this. So here they are in the lab. So they maintain this sort of wide mouth morphology in the lab. They do feed on scales in the lab, but they don't seem to be very good at it yet. And what Emily did is she looked at them, collected a whole bunch of these in the field before the pandemic. She looked at their diets, and indeed, they eat scales at about half the rate of the scale eaters. And all 4 of these species we collected were from the same lake, basically the same net halls. And indeed, their relative trophic position is isolated here in red, relative to generalists here in orange, and then the scale eaters in green. And so Emily then asked, why is novelty common within adaptive radiations, right? What's a potential mechanism for this? And so she looked at about 6.5 million SNPs across about 110 resequenced individuals of all 4 of these species. And the amazing thing about this system is that there are so few fixed SNPs. There's about 100 fixed SNPs in the scale eater for this lake population. But looking at these SNPs, Emily found that 15 fixed SNPs were fixed in both of these scale eating specialists. And essentially then she was able to time selective sweeps for all of the shared sweeps, as well as these unique selective sweeps in each of the specialists. And again, I'm showing this because this is wildly different from what we expected. What we expected is that first we'd see sweeping anything shared between the intermediate scale eater, this wide mouth, and desquamator. We thought, oh yeah, it's an intermediate, it's like a stepping stone. So we thought these shared sweeps would happen first in the intermediate scale eater, and then in the specialized scale eater. But what it looks like instead is that 5 out of 6 of the sweeps that are shared between these 2 specialists are happening first in the more specialized scale eater. And then later on, see these red sweeps of the same regions later on are happening in the intermediate specialist. So it looks like scale eating may have actually spread through gene flow from the specialized scale eater to this wide mouth species. And we're still sort of thinking about this, and Emily is going to talk a lot more about this in the spring. So in summary of just this first point, novelty is common within adaptive radiations and potentially due to some shared selective sweeps. So what about unique diet? We also think about the diets of these novel specialists as this is the only thing they're eating, right? But going back to Carl Lehm and his paradox is that specialists are simultaneously jacks of all trades. And so this is this classic paper by Lehm, as well as thinking about optimal foraging theory as a potential solution. But again, scale eating is only 50% of the diet of desquamator. It also eats macroalgae. It also eats snails. And my previous postdoc, Joe Aris, who just started his lab in this past year, 2020 at CSU San Bernardino, he was only in my lab in person for 3 months before the pandemic. But Joe actually looked at the microbiomes of scale eating specialists compared to these other species on San Salvador. And so he raised all of these fish in a common garden from 2 different lake populations, all 3 species, as well as some outgroups, and then looked at 16S sequencing of the gut microbial communities. And they're all fed the same pellet diet. None of these fish have eaten scales because we wanted to get essentially what's the genetic basis of any microbiome differences. But what we see overall is that scale eaters here in dark red, they're not that different from anything else, right? So trophic specialist microbiomes are just not that specialized. We did see, if you squint at this figure here, here's all the scale eaters that we sequenced here on the left versus everything else. There's a bit more red here. And so that red group is enriched in the scale eaters, even when they don't eat scales, is Berkeley, which is this group of bacteria that actually produce collagen, collagenase. So they digest collagen, which is the main component of scales. So we think that they're starting to adapt their microbiomes to digest their scale diet. My fantastic grad student, Jackie Galvez, in collaboration with a previous undergrad, Kira McLean, as well as Cyril Denning, who is my field guide and friend and longtime colleague in Cameroon and helped collect all these fish. We've also been looking at diets. And so Jackie, over the pandemic, analyzed all the dietary data from our Barrambian bow cichlid collections. And she found that indeed, here's pungu, they're the only species to eat sponges, right? But in general, if we look at their isotope enrichment, if we look at just the components of their diet across the board, here's pungu here and here's sponges, but otherwise they don't really stand out that much. So again, in Barrambian bow, we see that this unique diet, it's not immediately apparent. And so trophic specialists do appear to begin as jacks of all trades. So what about a unique environment, right? And I've talked about this before, but when we think about adaptive radiation, we think about these isolated large islands and archipelagos like the Galapagos Islands. But the pupfish do not follow this pattern, and they don't show a species area relationship like this. So from Massachusetts to Venezuela, effectively, here's the range of pupfish across the GBIF museum database. But now for the MBZ collection, we now have a great distributional range of pupfish as well. And as Michael mentioned, this is all basically due to Kelly Rice, who's this fantastic curatorial assistant I had for the past 2 years throughout the pandemic, who went and cataloged over 17,000 fish specimens in the MBZ collection. And of course, we had an advantage because we can catalog more than one individual essentially per lot, unlike the other collections, but also because fishes are awesome, right? And so essentially across this range of pupfish, we see most of them are essentially the same species with a wide range. They're allopatric generalist species that are somewhat difficult to tell apart. Here's some of that sort of diversity and reproductive coloration and black, fin patterning, but except in these two places. And they're both, pretty small islands. So one is the San Salvador Islands, it's only 10,000 years old, as well as Laguna Chichen Canab here in the center of the Yucatan. Both of these environments are large salty lakes. Both of them are quite large, about 20 kilometers, but they actually have almost identical competing fish communities, which is just a little mosquito fish in both places, and sometimes silver sides. So certainly this is part of the story of the origins of adaptive radiation, right? Is that you can't have really large predators or we don't get adaptive radiations. And I could stop there and say it's ecological opportunity, except that I've now surveyed all these neighboring islands, as well as some lab members that have surveyed additional islands. We've looked at hypersaline lakes. And essentially these lakes on the left that contain this trophic specialists and generalists look almost identical to the lakes on the right that just contain generalists. They're all these mangrove carbonate geology. And in fact, everything we've looked at now is comparable between neighboring Bahamian islands and San Salvador, including things now that we're looking at like macronutrients, lake productivity, isotopic diversity. The only difference we found are a few additional rare macroalgae species in some of the San Salvador lakes, and that might just be a sampling bias. So right now, major ecological differences do not seem to provide a sufficient explanation for the origins of adaptive radiation in this system. And we're sort of looking at these maybe more subtle differences. Specialists are jacks of all trades, and novelty is common within adaptive radiation. So that's to kind of summarize the ecology part of this. But next, moving on to behavior, right, is that we thought that this would be so simple, right? Scale eaters should be the most aggressive. And this actually goes back to this wonderful review of scale eating by Sazima in 1983, where he proposes that the origins of scale eating is due to more aggressive fish, right? It makes sense. And so Michelle took this on as the first chapter of her dissertation. And Michelle has this wonderful ability to take a simple project and then expand it and show us all the complexities, as well as this massive research program just based on this simple project. So what Michelle did was essentially the mirror test, which is used, essentially these fish will attack their own image in a mirror, and we can count the number of strikes and the latency to strikes over 5-minute intervals. And so doing this, she showed that the scale eaters actually have extensive sexual dimorphism in their aggressive attack rate, where the males are much more aggressive. But the same pattern, she found that the snail eaters, the molluscivores, are also more aggressive, which is not at all what we were expecting, and that it is less related to sex, suggesting that there's maybe a different mechanism here. And then she also looked at differential expression at early developmental stages and found, different sets of aggression-related genes are differentially expressed. And including some of these genes like MED12 is also involved in craniofacial formation. So potentially this aggression increased in the specialists could be a indirect result of selection on other traits. And so the story is much more complicated than we originally assumed. And so both specialists are increasing their aggression relative to the generalists, and potentially via different mechanisms. Next, Michelle took on another behavioral question, right? Is that during the strike, using our kinematic videos, scale eaters should open their jaws wide. This is such a simple hypothesis and prediction, right? And indeed, we can measure peak gape during those scale eating strikes that I've shown you already. And indeed, the scale eaters do have the largest gape at the sort of during their strike. But it turns out that they're actually, the generalists are opening their jaws more, resulting in a more acute lower jaw angle here relative to the scale eater. And so you can kind of see this here. They're feeding on gelatin cubes. And so here he's opening his jaw, removing a big bite of gelatin, which these fish love to eat. So Michelle sort of pioneered this approach of how do we measure scale eating ability and correlate that with the kinematics of feeding. Now here's this variegatus hybrid. And what Michelle showed then is that if these fish open their jaws too wide during the strike, they actually remove smaller bites out of that gelatin cube. So it really looks like there's potentially a performance peak for scale eating, and that these scale eaters have essentially adapted their kinematics to open their massive jaws less wide to get larger bites like this, which is one of the largest bites we've seen. And so scale eaters adapted kinematics to reduce their lower jaw opening during scale eating, the opposite of what we thought. And indeed, they can open their jaws larger if they want to. So they're essentially changing their behavior to not open their jaws quite as much. And Michelle's expanding this, which you'll hear more about in the spring, where to look across F2 hybrids and sort of really extensively measure what this performance landscape looks like for scale eating. Okay, so behavior, we thought it was simple. It's actually complex. What about genetic diversity here? And for genetic diversity, Emily Richards took this on for most of her dissertation, right? And her latest project, this is her 4th chapter, where she resequenced over 200 pupfish genomes and aligned them to our de novo assembly for the molluscivore, which is a pretty high quality assembly now. And now we have sampling from Massachusetts to California to Venezuela, so throughout the range of these pupfish. And first she showed what I would have sworn throughout my dissertation, that genetic diversity is just higher on San Salvador Island. And that's what explains this radiation there, because there's so many salt lakes, so there must be higher genetic diversity. But Emily has shown pretty conclusively here that genetic diversity on these 3 species that we find on San Salvador is not any higher than neighboring islands of Rum Cay or New Providence or Dominican Republic or North Carolina. And essentially we think that's due to extensive gene flow, due to things like hurricanes. These fish are all saltwater tolerant, so they are moving around among these islands. And next, Emily looked at genome-wide differentiation, and these specialists are somewhat diverged, but not that diverged, so about 0.1 to 0.3 FST. And so then she looked for these outlier SNPs, so the most divergent SNPs that are fixed or nearly fixed. It's only about 5,000 out of those 5 million that she looked at. And then she overlapped that with regions of the genome that look like they're under selection due to hard selective sweeps, using both linkage disequilibrium-based methods like Omega+, as well as site frequency spectrum-based methods like SWEDE. And so I call this the Emily triangle. But essentially, this was a set of candidate adaptive alleles that we think are involved in divergence of these trophic specialists. And then she looked at that set and looked at FD statistic outliers to test for introgression from different places in the Caribbean, as well as looking for adaptive alleles that were found everywhere, so essentially standing genetic variation, as well as alleles that are only found on San Salvador Island, de novo. And The shock here is that 98% of the adaptive alleles, so they're in hard sweeps, in the scale-eating specialists, exist elsewhere in the Caribbean. They're standing genetic variation or they've introgressed from a different source. Whereas for the molluscivore, 100% of these adaptive alleles exist elsewhere. They're not just found on San Salvador Island, which I really was just shocked to see. Only 2% of these are in coding regions, and many are in sort of cis-regulatory or intronic sort of near regulatory regions. And about half of these adaptive alleles are found in outgroup species, like Qualic here or Magupsalon, which last diverged from Cyprinodon roughly 5 million years ago. So an adaptive allele in a scale eater that's nearly fixed or fixed in scale eater, not found in a generalist in the same lake, is found in this highly divergent, isolated desert relic. We don't understand what's fully going on here yet. And so Emily also looked for, different sources throughout the Caribbean, and sort of she showed for this orange triangle here that introgression is not just coming from one place, it's coming from multiple places. And that's true for both of the specialists. Indeed, for the molluscivore here on Rum Cay, this neighboring island that also has salt lakes, it has all of the adaptive alleles that we see on San Salvador Island, but no molluscivore specialist. And some of this starts to make sense when we think about the geological history of the Caribbean. So about 10,000 years ago, here the landmass, the sea level was about 40 meters lower. And so the landmass of Nassau and many of these outgroups would have been the size of Hispaniola. So here in brown is this larger mass, whereas San Salvador Island was still tiny. It's just kind of a narrow pillar. And so we do see this signature of a sort of recent genetic bottleneck and lots more diversity on these neighboring islands. And we can also date the ages of these adaptive alleles from the surrounding genetic variation. And so here in gray is showing the age of the hypersaline lakes on San Salvador Island. 10,000 years ago, the sea levels would have gone down and all these lakes would have been dry. And so we see here on the right that most adaptive variation exists as standing genetic variation in this column, and most of it is much older than the age of the radiation, up to 60,000 years old and older. And so here for everything where it has an annotation, it's near a gene that's related to behavior or craniofacial development in blue, behavior in red, Emily has annotated that. But I also want to draw your attention to some of the de novo variation over here. And we have a few introgressed variation as well. But one of the last selective sweeps that did seem to evolve after the lakes formed in San Salvador, as we might hope, is twist one. And so twist one is actually one of the only non-synonymous substitutions that is in a hard selective sweep. So the structure of this, it's a transcription factor that's a basic helix loop helix structure. And right here, fixed in the scale eater, out of about 20 or 30 individuals that we've looked at so far, there's a proline to histidine substitution. And this substitution is actually conserved, so the proline amino acid is conserved across all vertebrates that we've looked at so far. All other teleost fishes have proline at this site, including humans, mouse, xenopus, all have proline at this site. And I know what you've all been waiting for is to see some mouse skulls, right? And so in knockout twist1 heterozygous mice, twist1 is involved in craniofacial formation. It's expressed in neural crest-derived mesenchyme. And in these knockouts, we see that it actually foreshortens the facial structure. So essentially, this is more brachycephalic here than this wild type mouse here. And twist1 is a key cancer gene. So involved in numerous different cancers. It's potentially involved in drug resistance to chemotherapy treatments, as well as stem cell formation in cancers, and is a potential therapeutic target. So it's just wild to me that we could learn something about cancer targets by studying a wild scale-eating pupfish, right? And of course, we want to work on CRISPR to do this. And so Joe McGurr is my first student to graduate from the lab in 2020, virtually, just like I'm giving the talk now. But this is Joe when he started doing CRISPR, the first CRISPR injections in pupfish in the lab, which is still an ongoing project. But Joe has also shown here that there are only 157 SNPs fixed between the specialists. And then he looked at structural variants as well, and there's actually 87 fixed deletions, all found in that scale eater. And so this just narrows the window down to looking for these candidate adaptive alleles and potential functional targets, right? And one of the best ones here, there's only a single deletion within an exon. It's the 5th exon of this gene, GPA33, which again, it's a cancer gene expressed in 95% of colon cancers, and it also has an unknown function. And so we'd love to know what this is doing in scale eater. And Joe also showed that with his expression studies, that 5th exon is not expressed across multiple populations, developmental stages, whereas is expressed the other exons. So in conclusion of this sort of novelty section is that novelty, rather than being all de novo variation like I would have assumed, it appears to originate mostly from ancient standing genetic variation, with some really standout exceptions like twist1, which we think might be kind of a refinement stage, refining that craniofacial anatomy. So what about gene expression then? We expected that these highly divergent trophic specialists should have different patterns of gene expression. And so Joe took this on in, I think this was one of his second chapter, where essentially he looked at generalist versus scale eater expression at early embryonic stages, as well as generalist versus snail eater expression. And so here, this larger circle in green are all the unique differentially expressed genes to this comparison. But this overlap here in blue are genes that are differentially expressed in both specialist comparisons to generalist. And out of those 818 genes, there's only 3, so all of them except 3 are expressed in the same direction in the specialists. So we think that this is essentially parallel expression in both of these trophic specialists, even though one is feeding on scales and one is feeding on mollusks. And when we looked at enrichment categories for these 2 different categories of parallel and divergent expression, it starts to make more sense. So what we see enriched for the overlap, the parallel expression, is actually metabolic process genes. So we think that they're sort of both adapting to more nitrogen in their diet. Whereas when we look at divergent expression profiles, they're enriched for craniofacial development as well as pigment biosynthesis, which makes sense since they're so divergent in that reproductive coloration. So instead of unique gene expression here, we see parallel gene expression in divergent specialists and much more parallel expression than expected by chance. There's a highly significant pattern there. The second part of Joe's work, which got even more surprising, is essentially for my whole dissertation, I would have said, yeah, these are young species. Genetic incompatibilities are probably not in play here. They're probably not important or the primary reproductive isolating barrier, which is still sort of a common view in some of the literature. And Joe first attacked this by looking at craniofacial tissues in 20 days post-fertilization in these larvae. And first, it looks like just wildly more genes are differentially expressed in these hybrids versus parental species. So essentially, this is what we mean by misexpression. These are genes that are differentially expressed in F1 hybrids relative to a pool of both sets of parents. And he compared that then to just generalist versus parent differential expression between these 2 species. But this is potentially an artifact. We had different RNA library prep methods. So Joe expanded on this at a different developmental stage, all new data, where he looked across 2 different lake populations and looked at differential expression first here in red between the 2 specialists, molluscivore and scale eaters, in both these lakes. And then in the F1 hybrids, he looked at misexpression in the F1 hybrids versus both sets of parents in both lakes here in blue. And so these are sort of the outliers in red and blue. But then he looked at the overlap here in purple. And so it turns out that these genes that are misexpressed in the hybrids, differentially expressed in the parents, are then enriched for all kinds of genes that we might expect, genes involved in craniofacial development, and a lot of sort of neuronal things, so things potentially involved in behavior. But this seemed to suggest that ecological divergence here might be causing gene misexpression as a consequence. And Joe also looked at a whole bunch of outgroup expression. So here on the right, he's showing FST divergence on the x-axis versus the proportion of misexpressed genes in these hybrid comparisons. And so if we just cross generalists from North Carolina to generalists from San Salvador, about 20% of the genes are misexpressed. And that's probably sort of geographic isolation by distance effects, maybe. But then if we look at Nassau versus San Salvador, almost 0 genes are misexpressed. So they're separated by about 300 kilometers here, right? Versus San Salvador Island, specialist crosses, we're seeing about 10% of genes that are misexpressed. So there doesn't seem to follow this sort of isolation by distance pattern that we might expect. And it seems like ecology is involved in this misexpression process. And indeed, for some of these genes, we see that in these hybrid specialist crosses, that they're expressed outside the range of any other of our populations, including these distant outgroups like North Carolina. So in conclusion of this little section, we think that misexpression might result from divergent ecological selection. But of course, we're excited to follow this up with essentially connecting misexpression to fitness. We don't know if this actually affects the fitness of these organisms, especially in the wild. But looking at sort of different misexpression hybrid families and comparing that to fitness in the wild would be a sort of fantastic follow-up to this. So the last section here is fitness. And most of this I presented before at MVC seminars. But starting these fitness experiments in the wild, I really thought that novelty should evolve from simple disruptive selection. And this idea actually goes all the way back to Rosenzweig, I think 1978, this idea of competitive speciation is that rare phenotypes within a population should have an advantage because of less competition. And this has been formalized since. It's a foundations of many speciation models. Essentially, negative frequency dependent disruptive selection can drive speciation because of selection for these rare phenotypes and essentially split a population. So when I started these experiments, I thought, oh, if I'm lucky, I'll get this nice quadratic curve and evidence for disruptive selection in the system, right? And so to do this, I raised over 2,000 hybrids, as I presented before, tagged them all with unique coded wire tags, photographed them all before release, bagged them up, put them into my checked baggage, brought them back to San Salvador Island, held them in these holding tanks, built field enclosures in these lakes. And finally, this moment of truth, reintroduced these F2 hybrids to each of 2 lakes from which their grandparents had originally been collected years earlier, right? And set up low and high density enclosures in these 2 different lake populations with all hybrids. Set them out in March, came back in July, they're still there, all right. And the environments inside look pretty similar to outside. Looked at all these 16 measurements of the pre-release hybrids, as well as populations of their parents. So all the measurements are occurring in a common garden raised in the lab. And finally, plotted all these measurements on a discriminant morphospace. So discriminating the 3 parental phenotypes. And essentially, this is a sort of a hybrid index, where within this space, we can plot the hybrids and their phenotypic similarity to each of the 3 parents. So here, generalists in blue in the middle, molluscivore down here, and scale eaters over here. And most of the loadings on these axes are essentially, as we move to the right along the x-axis, is an increase in jaw length. And as we move up on the y-axis, it's a decrease in nasal protrusion. And so here's the raw data. So these are all the survivors from one of those enclosures in black and all the deaths in gray. Of course, we can then fit a thin plate spline, and this complex surface was vastly more complex than I expected, right? And so that the generalist seems to be isolated on a fitness peak, separated by a higher fitness peak for the molluscivore. And the weirdest thing to me, though, thinking about this, is that these rare phenotypes over here, especially around the scale eater, they're not doing well at all. And so I actually repeated this whole experiment and tried to expand on this to really test for why don't rare phenotypes have an advantage here, like we expect. And so this time, as a postdoc now, this is 4,000 hybrids, fin clipped, frequency manipulated. This is in the basement of LSA. There's 1,000 fish in that tank, set up enclosures, again, larger enclosures. And I learned, just never repeat a good experiment, right? The first problem was only 3 out of the 4 crates made it to San Salvador. One of them got lost in the airport at SFO. Then a hurricane, my namesake, came towards the islands after I'd set up the experiments, but it veered away at the last minute. But I ended up collecting them too early. And the second set of enclosures I left out there for too long, a full year. And I'm just kind of amazed that they were even still out there when I went to collect them. But then as I started my lab, I had this amazing lab manager, Kate Gould. She's now a grad student at UNC, and she sorted through all this mess of an experiment. So she read all the tags, she did some DNA extractions, and even that alone basically took a year. And what we found after all of that work, so this was just published last year, but essentially the top row is Lake 1, including a control of these hybrids raised in the lab over the same time period. And the bottom row is Lake 2. with that control. And then this frequency manipulation was essentially moving these very weird extreme phenotypes into one enclosure and taking them out of the other. And there was a sort of significant difference in phenotypic variance between these two enclosures. But what I found after all this is that it had no effect. This treatment effect had no effect on differences here or here. And so these rare phenotypes, if you change their frequency, it doesn't seem to affect the fitness landscape. But what we did find is that in the first lake, of course, these lakes are actually quite different this time, but we see that the generalists and the molluscivore here, hybrids resembling those phenotypes, seem to do pretty well. And then in the second lake, so that's about the short term, about a 3-month exposure period. In this bottom row was a full year. So in the long term, almost essentially all the survivors, and there's very few, but they all resembled the molluscivore phenotype. So in the long term, these molluscivores do really well. And using general additive modeling, I was able to basically estimate one surface using the best model for describing these experiments. So using model selection and then visualizing what that sort of unified fitness landscape looks like across all 4 of these field enclosures. And that landscape is this right here. So in both 2 dimensions and 3 dimensions, when we combine these lakes and estimates, there seems to be generalists on an isolated fitness peak again, as well as the molluscivores on this higher peak. And it just, to me, is surprisingly similar to the original experiment, where we expect that fitness is this stochastic thing, that these landscapes are changing like the surface of the ocean. But in fact, for these really highly novel divergent specialists, there might be more stability to these macroevolutionary types of fitness landscapes than we might expect. So the fitness landscape underlying novelty is surprisingly complex and surprisingly stable. So in the last 4 minutes or so, what we'll hear about soon is my postdoc, Austin Patton. And what Austin wanted to look at is novelty isolated on the adaptive landscape, which this original idea of adaptive landscape goes back to Wright in 1932. So these are actually genotypic fitness networks where we can see 5 different loci moving from the wild type to fixed for all these minor alleles here. And we don't have many examples of this for vertebrates. So what Austin did, he's only been in lab in person now for 2 months, but he's been a virtual postdoc now for a full year as the pandemic started. And he's also has the distinction of working on all major groups of vertebrates. But we resequenced 142 hybrids from those field experiments. And showing you here, there's lots of diversity in sort of the ancestry of those 3 different parental species. And Austin showed that he could find a genetic basis, so SNPs associated with improvements in fitness here. And when he includes those in models estimating that fitness landscape, it actually improves inference of the fitness landscape. Even with about 150 fish, he's able to sort of pull out that generalist peak again and a sort of a ridge valley separating the molluscivore, as well as this really deep trough for the scale eater. And let's see, I'll go through this pretty quick here. Let me cut through to the end here. What Austin did, which he'll tell in a few weeks, and it's wildly complicated, so I think I'm going to let him explain, is that essentially about 2% of genotypic pathways between the generalist and scale eater are still accessible. So even though everything I've shown you so far suggests these scale eaters are in a fitness valley, at the genotypic level, we are still finding that there are pathways through that space where you can increase your fitness at each step. And that to me is a surprise. And so there are these rare accessible genotypic pathways to scale eater phenotypes. So of course, we're trying to put all of this together. We're trying to build this up to discover new craniofacial genes in pupfishes. My postdoc Fania is going to start on this in just a couple weeks, and she's here today. And so Joe has started to do this, as well as Michelle has 2 different genetic mapping crosses where we've crossed the specialists and look for shared QTL among the lakes. And we also have some cis-regulatory candidates where genes that look like they're cis-regulated based on F1 hybrid expression patterns. One of the best ones is PICER3, where we actually have a knockout of a zinc finger binding motif here. And we've even started to do in situ hybridization, looking for PICER3, which has never been shown to be expressed in craniofacial tissues before. But here, these little brown dots are the transcript expression patterns from RNAscope, where we think that it's expressed in the neurocranium as well as some of the jaw tissue. So how does novelty evolve, right? It's common during adaptive radiation. Ecological opportunity is not a sufficient explanation. Scale eating is not as simple as just maximum aggression and gait width. Genomics is that mostly ancient genetic variation appears to be reassembled into new adaptive combinations to build these trophic specialists. And that some of these specialists actually share selective sweeps and metabolic gene expression as they adapt to divergent trophic niches. And finally, the fitness landscape is stable with isolated fitness peaks, but rare accessible genotypic pathways. And last, I don't want to leave out David, my fantastic grad student who's been working on the Devil's Hole pupfish, as you've all heard about at the MBZ symposium. But to come back to the beginning, I mean, David was able to successfully extract DNA from museum specimens. Formalin-fixed Devil's Hole pupfish are critically endangered species, as well as identify deleterious mutations and knockouts, with the potential for actually what we hope to do one day is genetic restoration. So who knows what will happen in the next 10 years, right? And with that, I want to thank my wonderful lab, past and present. Here's many of the past lab members and funding sources. In particular, NSF, NIH, all the kinematics work is Binational Science Foundation. All of our Devil's Hole pupfish work is US Fish and Wildlife and National Park Service. And thank you all for your attention.
Thank you, Chris. Please join me in unmuting and thanking Chris for a wonderful talk. I was delighted to see that you worked in a mouse skull into your talk about fish there.
I'm glad to hear that.
So let's open it up for questions. Go ahead if you have a question. You can unmute yourself and ask away.
Yeah, so Mallory, that's a great question. And in fact, I think in the audience, I have Hwan Chan, who's just a fantastic undergrad that took my Ichthyology class virtually last year. But Mallory asked, have you measured the role of phenotypic plasticity in generating pupfish traits? And so superficially, we've raised these fish multiple generations in the lab. Most of that morphology is heritable. But there's always some plasticity involved too. And Hwan's sort of senior thesis project is actually to try to feed some of these scale eater fry scales in the lab using things like sliver sides and compare those to pellet foods. But then he brings in this brilliant idea of essentially looking at the vector of change for some of these craniofacial chains and aligning that to his work that he's already done, looking at CT scans, because everything had to be virtual. Where he looked at CT scans of 4 different families that contain scale eaters and looked at sort of convergent evolution of scale eating craniofacial traits. So I'm excited to see what he finds.
We've got a question from Jim asking how easy is it to successfully cross these species, from Jim.
Yeah, that's a great question too. I mean, our Barombian bow cichlids, we've been trying to cross now for 10 years and it's happened once and only with 2 really closely related species. But the pupfish species, which I assume you're talking about, are much easier, but we essentially don't give them a choice. We just have a pair within a tank, and usually within about sometimes just a couple weeks even, we're getting eggs. Some of the other pupfish species that we've crossed, like the ones from Chichen Kanab, will take 9 months. I don't know why, but we'll have them together and they just won't lay eggs, and finally they do. And so it does vary pretty widely, but it's a great system because we can cross them so easily in general.
Lawrence, you've got your hand up.
Thanks, Michael. Hi, Chris. Thanks for that overview of the great work done by folks in your lab over the years. With that in mind, I wanted to ask you something a little bit different. I want to ask what your general mentoring philosophy is and how you've been able to best help your students succeed and do this work.
Yeah, I don't know, Lawrence. Ask me in 20 years how it's going. I think we're all, I don't feel like I'll know how I am as a mentor until I can see my students, eventually get tenure and far out, right? That's one answer. I think, we might have kind of a flipped philosophy sometimes where, especially with the pandemic, or when students are often, most people, they join the lab and they do a first year project, but in some sense before they go to the field. And and eventually my goal is to get everybody to the field. But with Cameroon and the pandemic, that's been hard for sort of the Cameroon studies. But yeah, I mean, my mentoring philosophy is just open door policy and to have people come and talk to me whenever they want and try to meet with everybody in the lab once a week, but sometimes that falls through the cracks. And just to try to help, people achieve their goals, right? I don't know if that answers your question, Lawrence. Did you have something more specific in mind, or?
No, there are lots of other questions in the queue, but thanks for that, Chris.
Okay.
Ben is asking, can you explain how you estimated the timing of a single SNP?
Yeah, and so you will hear much more about this from Emily's exit seminar in the spring. And what Emily has been doing, I think her most sophisticated approach is the STAR TMRCA method, which uses site frequency spectra, as well as linkage disequilibrium patterns to estimate the timing when we have sufficient sort of lots of resequencing. But initially, we were doing we do need a mutation rate. So we actually have estimated the pedigrees of these pupfish populations, where we have 2 different pedigrees now, where we've sampled them to high coverage, sequenced both parents as well as multiple offspring to try to estimate a mutation rate for Caribbean pupfish, which is a little bit higher, which we think is probably because pupfish have a higher metabolism. And essentially then with that mutation rate, we can look at essentially just background genetic mutations within these regions of the sweeps that we're interested in, as sort of a more sort of old school or low tech way to date these sweeps. So we've used both approaches.
Rory, I think you were next.
Oh, thanks for that great talk, Chris. I have a whole bunch of questions, but I'll see if I can get them all out quickly.
Sure.
So the first kind of question is, so I was interested, I think the work you've been doing on these adaptive radiation is fascinating, but I was struck by, the number of fish that you've curated and just showing their distribution map of the wide distribution. And so that sort of raised 2 questions for me. One is, have you collected sequence data across a larger evolutionary spectrum? And I'm really curious about the role of gene flow more broadly across the phylogeny for these fish. And like thinking about how to collect or connect sort of the microevolutionary processes with the sort of macroevolutionary processes. And, I imagine there's a lot of cool things you're thinking about doing with that. So that's one question. And then the other is, I'm always struck when I see the picture of the devil's hole pupfish, how blue it is. What do you know about the pigment genes in these fish, or how these pigments are generated? That seems sort of I haven't seen much published on that, and I'm curious about it.
Yeah, 2 awesome questions. And so Rory, I have sequenced a whole bunch of outgroups across Cyprinodon, as well as even further out across Cyprinodontiformes. I mean, the goal is actually to get a complete phylogeny for Cyprinodon today, which I think we have most of the genera sequenced now. I haven't had anybody to put it together yet, which is kind of a lame excuse, but it's there sitting on our server. We have all the genera sequenced. And I think it's a great question too. And I think, something that Austin is interested in as well is just looking at these patterns of introgression through time. I mean, particularly with these ancient alleles in the Goopsalon and Qualic, and trying to trace those gene trees back even earlier has always been, I think, as soon as Emily's work came out with that, I think it's been a couple years now where that's been fermenting, but just thinking about those gene trees and how do we even quantify introgression across, 20 million years in some sort of meaningful way, I think is also tough, but and I'm sure you're thinking about similar questions. But yeah, it's a fantastic question. We do want to do that. And I think there's a lot of really interesting things we could do with looking at sort of more macroevolutionary patterns and where do these adaptive alleles come from. And then your second question, so yes, devil's hole pupfish is more blue, like a darker blue than any of the other Death Valley pupfishes. And no one has looked at that. But David is now sort of working on a massive project where we've just sent out about 100 samples for sequencing, 100 individuals, and then he just brought back 100 museum fin clips from devil's hole pupfish from Reno. So we're trying to get historical sequencing as well, so that we'll actually have a large number of genomes to say what is unique to devil's hole pupfish. But so far, with this sort of pilot project, this awesome project that he's done, we think that there's essentially so much drift, and it's such a large sort of small population on a large bottleneck that we might not even be able to detect selective sweeps or even have much evidence to show that something is adaptive. I don't think he's found anything related to pigment yet, but there's probably something there. And we haven't really focused on pigment yet. Great, thanks.
David Mindell asks, how would you define evolutionary novelty now in light of your cool findings?
Thanks, David. Yeah, I think I love this old paper by Gary Vermeer where he says, all these ancient novelties that we really think about, we only have one example of. But all the recent examples of novelty, there's multiple examples. And he proposes, and I think he's right, that this is essentially just a filtering effect, right? Where we're missing, essentially over time, only one of the novelties survives, right? And we're missing all of these sort of early examples of novelty. So I would sort of cast a broader net now as we look for novelty and trying to capture evolution of novelty in action. But I do still wonder about, what are the microevolutionary processes surrounding novelty that might be different from local adaptation, right? Like I didn't talk at all today about differences among lakes between the same species, which we do see. The molluscivore in particular has sort of different nasal protrusion in different lakes. And thinking about how do patterns of local adaptation, how does adaptation differ across novelty versus just sort of local differences in clines? And I think it's still an open question.
Ammon, I think you're next.
Hi, Chris. That was a great talk. I was really impressed by your fitness landscapes. But one of the things I was wondering was, given the low, it seems like the relatively low fitness of the scale eaters, how do you think that they're being actually maintained in the environment, because they're actually this morph out there? And I know that in some scale eaters, there's some that are like handed in different environments because there's frequency dependence about like other fish looking out for them. And so I'm wondering if there's something about like a small enclosure that maybe all the other individuals learn who's the bad one who's eating their scales, but I'd love to hear your thoughts about like, what do you think's going on there?
No, both fantastic questions. And I do think that so that scale eater fitness peak, I think it's out there. It's kind of my white whale now, because we still have no evidence from growth rate, from survival, that there's anything like a peak in that region. And I do think one reason, even that's why I built the enclosures larger the second time, was this idea that maybe the scale eaters need a really large range. So I'd love to be able to tag them and track their range in the wild, which we can't do because these RIFID tags are just not quite small enough yet, at least on the commercial market. But a sort of argument against that is I also kind of thought, well, I at least should get one scale eater hybrid phenotype just doing really well, right? And we don't even find that. We don't even find one. And so, is their home range really that large? Certainly in the wild, they're maybe every meter or so, like square meter. And the females are ranging, but then the males are guarding territories. So I don't think they have a massive home range, but it's always possible. And then another reason is just I've only used hybrids so far. So you might need all these different sets of metabolism genes and kinematic, the right coordinated kinematic response to be a successful scale eater. So essentially that fitness peak might be just very narrow and very hard to access. And so that, we should definitely do the experiments where we put out the sort of purebred scale eaters raised in the lab. But even then, maybe if they're not eating scales their whole lifetime in development, maybe they're still not going to be that adapted in these field enclosures. And then let's see. Yeah, that was your second point too, right? Yeah, no, great question.
Yeah, that's right.
Thanks.
Thanks so much.
Thanks, Ammon. I had the same question. Craig, it looks like you're next.
Great talk, Chris. I'm wondering about the distribution of adaptive variants in the pupfish genome. I was surprised to read in one of Joe's recent papers that inversions don't seem to be common or found, but I'm wondering if the adaptive SNPs are non-randomly distributed. Are there pupfish supergenes?
Yeah, that's a great question too. And so I think we have one or two QTL now. It's not nearly as cool as the sort of stickleback story, right? We have one or two QTL where they seem to be affecting multiple craniofacial genes. And indeed, across different lakes, Michelle has found that, depending on how you count, there might be 7 out of the 8 QTL we detected that are affecting the sort of same sort of integrated craniofacial trait complex. The reviewers didn't like that. They said we should just say there's 1 out of 8 shared. So I think in terms of the distribution of these adaptive regions, and to address your question about inversions, so Joe has only used DELI, we've only used short read sequencing so far to look for inversions. But at the same time, when we look at sort of genome-wide FST profile plots, we don't see signatures of inversions, right? We don't see huge patterns of high LD. And so it doesn't look, we certainly could be missing it, right? In terms of we need a better assembly, we need long reads, which is certainly we'd love to do. But so far, we don't seem to see much evidence of inversions, and I'm not quite sure why. And there is this sort of interesting contrast in the literature right now, right? Like sunflowers too, and all these other systems where inversions are so important. And I'd love to have more inversion, although, in some sense I wouldn't too, because it's much harder to map within an inversion, right? But we're, definitely on our radar, but I think we just haven't found it yet. And in terms of, I think we also need to look at many more traits like you guys have done with stickleback, right? I love to look at gill rakers and see if there's different QTL controlling different gill raker lengths and things, but lots more to do. Thanks, Greg.
Well, Chris, that was just a fantastic talk. You are truly an integrative biologist. And it was so fun to hear about all the things that you and folks in your lab have been doing. So thanks very much. And to everyone else, you know where to find Chris. If you have more questions, he's around. So thank you, everyone. And we'll see you next week.